Hi everyone and welcome to today's episode. My name is Antony Jose and I am pursuing my undergraduate in biotechnology with a focus on vaccine development. Today we are diving into a fascinating topic of how machine learning and AI are transforming the field of biology, and in this episode we'll particularly focus on a generative model that is variational autoencoders and its use in drug discovery. Drug discovery is an extremely long and expensive process that can take over a decade and cost billions of dollars, and even then, most of the potential drugs could fail before reaching the patients. But AI and machine learning are now changing this, cutting down cost discovery time from years to months. But how exactly? If you look at the traditional drug discovery process, scientists need to identify a biological target, like, say, a protein which is linked to a particular disease. And this is then achieved by extensive literature review. But if it is a novel pathogen, we need to build our knowledge from the ground up by studying the pathogen, how it is interacting with the host, etc. And after a target has been identified, it is then followed by screening of thousands, sometimes millions of chemical compounds against the target to find a promising drug candidate, which is then followed by selection of some of the promising candidates. Refining the best candidates can itself take years of lab testing and clinical trials, and most of them might fail in the final stages. As it is evident here, this is a time and a money consuming process. And this is where AI and machine learning can help us. AI can analyze vast amounts of biological data and identify the most promising drug targets faster. That is, it will be helping us understand disease biology. But not only that, instead of screening molecules randomly, machine learning models can generate and optimize potential drugs in a more efficient way, that is, speeding up the compound design, and it can also help us predict which drug candidates are most likely to fail, reducing wasted resources and increases the chances of success in clinical trials. We wouldn't have to like waste so much time screening drugs which are probably going to fail. And one of the most exciting AI driven innovations in drug discovery here is the generative models itself. They allow us to design new drug like molecules in a virtual environment and then testing them in a real lab world experiments. This approach significantly cuts down on time and resources, and that would normally take months in a wet lab can now be done in seconds, yeah, hours using computational tools. The core idea behind generative model is a three steps cycle of generate, score, and select. AI models create new molecular structures, and then these molecules are evaluated based on important parameters like ADME, binding activity, selectivity, and ease of synthesis. And then from these, the best molecules are chosen, and the process is then repeated until we find the most promising candidates. One of the most generate one of the most powerful models, like among many others, like we have a lot of generative models like GANs, GNNs, RNNs. So from these, one of the most powerful ones is variational autoencoder. Unlike traditional drug discovery methods that rely heavily on trial and error, VAEs, that is variational autoencoders, they intelligently explore the chemical space, identifying promising drug candidates faster and with greater precision. But before we get into variational autoencoders, first, what is an autoencoder itself? To understand variational autoencoders, let's understand autoencoder properly. An autoencoder is a type of a neural network designed to learn efficient representations of data. I'll explain that a bit. Like the autoencoder has two parts, an encoder and a decoder. The encoder will take input data. The input data can be anything like images, text, or in our case, molecular structures, and compress it into a smaller fixed size representation. And the decoder will then take this compressed representation and then try to construct the original data as accurately as possible. The goal here is to minimize the reconstruction log, reconstruction loss, meaning the decoded output should be as close to the original input as possible. And when this is applied in drug discoveries, molecules here are represented in formats like smile strings or molecular graph, and then they are fed into the encoder. The encoder compresses these molecules into a fixed code, into a latent space. It is a mathematical space where similar molecules are grouped together. The decoder then attempts to reconstruct the original data from this latent space representation. I'll get into the latent space in more detail. But first, basically, what a standard autoencoder does is it memorizes specific mapping rules for each molecules, which makes it good for compressing and reconstructing data. But it has a major limitation. It cannot generate any new molecules from scratch. It only learns how to reproduce what it has already seen, and therefore they are not much useful in drug discovery. So coming back to latent space, what is a latent space? Like we talked about how the encoder is compressing the molecular graphs of molecules into a code in the latent space. So a latent space, imagine, look at it like a 2D map of molecules in which it's basically a 2D map of molecules in which the similar molecules are positioned close together, forming clustered based on shared properties. Each axis in this space can represent a different molecular feature like size, solubility, or molecular weight. And moving along an axis allows us to tweak these properties and generate variations of existing molecules. For example, let's say we start with aspirin. Moving along one axis in the latent space might result in a stronger painkiller, while moving in another direction could create a version with fewer side effects. This structured latent space allows us for fine tuning of molecular properties in a way that traditional methods cannot. What is a variational autoencoder? Now, coming back to our main topic, a VAE is an improved version of an autoencoder that introduces probabilistic modeling. That is, instead of mapping each molecule into a fixed compressed code, a VAE represents each molecule using a probability distribution of a mean and a variance. Here the mean represents the most likely location of a molecule in the latent space, and the variance represents the uncertainty or how spread out the molecule's position could be, and that will help us in creating more variations of the molecule. Instead of encoding a molecule into a single point in the latent space, the VAE samples points from the probabilistic distribution, and this introduces slight variations, allowing us to create new molecules rather than just memorizing existing ones. That is what an autoencoder does, but not a variational autoencoders. It can create new molecules from existing ones. How does a VAE work in drug discovery now? Like how is it applied? How is the process actually? It has two main phases, we can say generally, the training phase and a testing phase. In the training phase, the VAE is trained on a dataset of molecules, gradually learning to create a well organized latent space where similar molecules are clustered together. And during the training, the decoder output is compared to the original output, and two types of loss are minimized. First is reconstruction loss. This ensures that the decoded molecule is as close as possible to the original molecule. And the second is regularization loss. This ensures that the latent space follows a smooth and a structured distribution, and this prevents overfitting and ensuring that all the points correspond to realistic models. Yeah. And so after the VAE is trained, we can now move on to the next phase, which is the testing phase. Once trained, the VAE can generate completely new molecules by sampling points from the latent space and then decoding them into a valid chemical structures. Scientists can now modify the molecular properties by moving along specific axes in the latent space and then effectively fine tuning new drug candidates. For example, in the training phase, if we want a molecule with similar binding properties to an existing drug but with a fewer toxic side effects, we can explore the latent space to find a promising alternative. Why are what are some of the advantages you of VAEs in drug discovery, like when we compare it with other generative models or even like traditional drug discovery processes? First is VAE can generate a wide range of novel molecules, increasing the chances of finding breakthrough drugs. And then one more important one here is like the scientists can manipulate molecular features like solubility, toxicity, and binding affinity in a controlled manner. Like that is what I talked about just above right now. And another one is VAE can significantly reduce the need for costly and time consuming process, which we usually see in traditional drug discoveries, and allow for in silico testing. And then we can check the best candidates for real world validation. But of course, the VAE also has some challenges and limitations. If we are to look into it, like not all the generated molecules may be synthesizable in the lab, and but one advantage here is that graph-based VAEs can help address this issue by preserving the molecular structures more effectively. One more limitation here is that there is bias in training data, because if the training data is biased or if the training data is limited, then the generated molecules may also be a reflection of that bias or that limitation of the training data. But that can also be overcome easily if we are like if we have a wide variety of data sets or a wide range of data sets. Another one is understanding how the latent space encodes specific molecular properties remains an ongoing challenge which is still being explored till date. Finally, if we look at this, variational autoencoders are recently revolutionizing the drug discovery by enabling the generation of novel, high quality drug candidates at unprecedented speeds. And it's not just variational autoencoders, AI and machine learning and n number of generative models that keep on emerging, or new modifications. Even VAE has a lot of modifications. Every now and then we see a new variation of the previously existing models coming up. And while challenges remain, continuous advancements are still happening. VAEs here are they are used in pharmaceutical research extensively, and yeah, by integrating them with other computational techniques of like molecular docking and simulations, the future of drug discovery is set to become faster, cheaper, and more efficient, and ultimately lead to better treatments for diseases worldwide. Yeah, thank you for this episode.